Sure
Okay,
it's videoing this time.
I think we have a video. Hello, everybody, and welcome to episode 1 of a series. Today we have Mr. Samuel O'Donnell joining us today for a chat about his upcoming science research and PhD and general science talk.
Give it a couple years before you Google me. That's all.
Yeah, okay.
I don't think there's much.
So,
There's
yeah,
much there
look
at the
out
moment.
for Sam the scientist.
Yeah. I think I'll go with that
In the future.
tagline.
So, you are about to start your PhD next week in France, leaving New Zealand.
Yeah.
Looking at what we both have a history of working in molecular biology work, looking at DNA. So, what exactly will you be covering?
It's a bit different though, eh? So as I've been working on plants previously, I'll be jumping into yeast, and this one, although I'm jumping into a larger project, which is looking at how yeast function in the environment, and we're going to be looking at these mutations.
So yeast are what are used to ferment grapes, and they're the white
bread
and used in making bread. Okay, so that's how most people would.
Yeah, so a lot of these
yeast.
food processes, these fermentation processes, are using this Saccharomyces cerevisiae, which is the main species. Although you're going to find that there's a lot of variations in it. So the one they use for bread might be the same species as what they use for wine, but they're not the same. So that one that they use for wine has been cultivated, has been processed,
sort of been selected
for
for the job.
But that's a very small subset though. So the group I'm working in has gone around the world, sampled all of these different variations on the species, and they've now got this database of 1,000 or so different variations of the species.
All genome sequenced.
Yeah, rough genome sequencing. And what?
How big's the genome? Do you know?
I looked it up. I think it was
100 megabases or something?
Yeah, I think that is what it was.
Okay.
Or 30 megabases.
Right. We'll check that.
Yeah, don't.
And
don't quote me on that one.
Right. So you're using yeast as a model for mutation rates, like to look at evolution?
Yeah, so the initial project is looking at these large mutations, which they're called structural variations. So previously, all these genome sequences allow you to look at these smaller base point mutations.
Single changes.
Yeah, and the reason why you can't look at these structural variations very well is because the technology used to sequence the genome puts out little sections which you have to paste together to create a larger length.
Right.
And these new technologies allow you to create these larger lengths in one go. So you can see these
so you
larger
sequence
changes.
larger fragments. Okay.
And then you can see larger fragments that have changed between different different variants of the species. And so that's the initial project, and we want to look at what these changes do to the species in the environment. So these ones that are fermenting bread, or these ones that are fermenting grapes, is there any structural variations which are determinant for this ability to, act on different metabolites, different areas, live in different places.
Right, so that's interesting in how it could possibly be affecting what it does and what environment it can survive in. But also, has this been used at a larger scale for determining the rate of evolution, like the molecular clock?
Yeah, so that's my side. Is that with all this data, what has been coming out recently is that instead of using this molecular clock, which is determining rates of change based on mutation, looking at different points in a genome.
So just to cover the molecular clock a little bit, so the idea is that you can determine how far two species are apart, or how long it's been since some ancestor, by looking at the genome and figuring out how many mutations have occurred, assuming some constant rate of mutation, which is safe to do over large timescales, so tens of millions to billions of years. It does kind of even out to some constant mutation rate.
Yeah.
So I think that's the idea behind the molecular clock.
And so that's what's used in terms of phylogenetics. And so looking at family trees, what we're using there is a molecular clock to tell which parts of the genome, and they're selecting certain parts, say certain genes, which are of interest. and looking at how similar they are and what's changed, and so where the progression is of change. But what's coming out now is that this may not be the most correct way to measure it. It is still a way of measuring relationships, but it may be that this change, this genetic change, the mutation rate may vary completely along a genome. And so the next step is calculating the genomic clock. And so we're going to be using these structural variations.
The genomic clock, I like it.
Yeah. So why these structural variations are important is because the genomic clock may change just based on how similar two pairs of chromosomes are to each other. So when we have sexual reproduction and you have from your mother to your father, based on how similar they are to each other, may be a determining factor on how much or how high or how low the mutation rate is in certain parts.
Is this kind of opposite to inbreeding? Because if you inbreed, you think that normally there's more problems occur in the offspring because the DNA is too similar between the...
Yeah, I'm not too sure how it really adds into the inbreeding idea.
So, but for yeast, if they're very similar, is the mutation rate higher or lower?
The idea is that that's going to be lower.
Okay.
So how the greater difference is most likely going to have a greater rate of mutation, and so these structural variations, we want to see if when these structural variations occur, possibly on the flanking regions, because the difference has become greater because this is a new mutation, so it should be greater because the gene pool shouldn't have this new mutation. We should see more mutations lining up along the sides of it.
So you might find like islands of mutation.
Yeah, so we get these big structural changes, and then all of a sudden flanking regions and that become far more susceptible to mutation. And this is based on what is the actual functioning part which determines it isn't known, but we're going to be just looking at it in terms of similarity and how that affects mutation rate. But the idea is that it's because of the machinery that moves along and it affects that somehow, and this is how mutation rates change in that area.
Very cool project. I like how it can apply real broadly, and I would be excited to see comparing the molecular clock to the genetic clock and seeing if it's just higher resolution of the same thing, or if it gets different ages.
That's one of the things, is it's a bit of a textbook changer in terms of phylogenetics, because if this is applicable to the same trees, then yeah, it'd be nice to see whether we've been on the same track in everything, or whether in some parts maybe it's we're going to be filling in some clues there.
Right, yeah. So it's interesting, all these new technologies coming out. So that's a cool project, very bioinformatic, very sequencing heavy. And with all these other new technologies which allow researchers to just play with DNA and read DNA and change it, and like CRISPR-Cas, with very specific, precise edits of the genome, it's getting quite impressive what people are able to do. We're getting to the stage where we can just synthesize DNA, so Craig Venter has made his synthetic genome.
Yeah,
so
he's on track.
People are just going to start printing out genomes soon. You'll be able to print a genome for a person, and that's getting quite amazing. How do you think the race between that sort of technology versus artificial intelligence, who do you think is going to win out of that? Because ideally, I guess that they're working towards the same thing, as like stopping us from dying, like what medicine does, I suppose, or making us smarter. What do you think?
Yeah, who's going to win in
that? Who's going to win? Yeah.
So I think in terms of that, they're both just trying enhancements, whether it's enhancements in terms of productivity or enhancements in terms of longevity of life. So maybe in that sense, it's that different areas would be more susceptible to whether it's human biology and that pathway, or cyborg AI pathways. But why I find the human biology one a more realistic, or at least socially acceptable format, is that I can see it happening in a more small incremental changes in terms of changing an enzyme, reverting mutations, stuff like that.
So that already exists, right? People,
Yeah.
they get fertilized human embryos and will remove a genetic disease, or
So
starting to be able to pick up on
gene
stuff like
therapy
that. Gene therapy
is working.
Cancer treatment tries to do things like that.
Yeah, gene therapy.
So
again.
in that way, it's already accepted, whereas maybe robots will struggle more. Do you think?
Well,
Japan's
I don't
pretty
know
good at robots.
I think we've already accepted robots in that sense too, though, because we're replacing organs with we've got pacemakers now. So there's that fighting disease avenue where both are working on it. So maybe they're both already there.
Yeah, so both exist, but I guess one of them's going to win, because once we get one, I guess if we cure ourselves biologically, we'll still end up with robots. But if we get AI robots first, we might not ever cure ourselves biologically, because we might just move to the robot.
I don't know if
you get your cyborg arm, and then your cyborg hip, and then your pacemaker, and your cyborg heart. And
I don't know if they're so separate in that sense though too, because with proteins now, one area is actual artificial proteins, and so that's adding parts that aren't available in nature into a protein so that you can make it function in a completely new way. And it's possible that these two avenues might just meet in terms of not creating a whole arm that may be robotic, but creating maybe something more interior, so that we don't feel like we've lost our body, but we're still biological physically, but we've just got these smaller enhancements which are maybe less
scaring
yeah
of the people.
I think it's
because people are scared of stuff, but it's already, you can sort of see where it's coming from. Like, we're already cyborgs in a sense, because we've got our phones in our pocket. These are supercomputers. We ask it all the questions we don't know the answer to. It knows where we are. It gives us directions. And then it's in your glasses with Google Glass, and then it's in your contact lens maybe next, and then you might just end up with some brain implant where you got Google in your head. And where will people draw the line?
Quite true. I don't know where they draw the line. Where maybe the scariest part is the whole what makes you a person, and whether if you were to remove the brain and put it into a completely robotic
the personal
body,
identity thing,
is that still you? And as soon as you remove it, who are you afterwards?
Yeah. The brain damaged patients and like brain surgery stuff must shed light on the personal identity stuff, because they do I don't know what it is when they sever your hemispheres. What's that joining part? I forget the name. And then you can almost have arguments with yourself, and then you get like those mirror agnosia patients who ignore one side of their body and things like that. So
the ghost arms and that, the
phantom limbs and stuff.
Limbs,
where you get an itch on your So that stuff's quite amazing. And then so the prosthetic arms and stuff are getting pretty good now as well.
Yeah, those ones, that sort of idea links in terms of adding beyond the human form.
So not just repair, but improvement.
Yeah, but completely beyond it. So it's not just making this arm stronger, it's adding another limb itself, it's adding other legs. I feel like the limbs are the way to start. I don't know what the bigger idea would be, but it would be to create a whole nother limb, and our brains are malleable enough that it's possible. As you see with monkeys using electronic arms to feed themselves without having to move any other limb.
Yeah, so controlling things with mind power.
And
that's
then
cool
that's the
technology.
prosthetics. But it's just, instead of replacing that arm, it's just giving you another one.
Yeah, so you could. It doesn't have to be another arm. You could have a robot following you that you're controlling with your mind as well, right?
Yeah, it doesn't actually have to be attached to you.
You're like
at
a
that
hive
point.
person, yeah.
Yeah, but that's all the
That's
nanobot
crazy
stuff. Yeah, I don't even know if I've thought about that actually.
Yeah, that's crazy stuff. So it's interesting. Like, I hope we go down the biological route, and because that's what medicine has been so good at so far. We've pretty much solved infectious disease, except for the pro-disease movement, which calls themselves anti-vaxxers,
No.
and so there's starting to be a little resurgence in essentially cured diseases, but where we haven't made much ground is in things like cancer and Alzheimer's and these diseases of old age. So it'll be interesting to see how this type of research, learning the DNA, how it behaves, will feed into that, and we might see some impressive gains.
Yeah, I guess it's the reason we see that in terms of life is that the reason we were dying so early was these infectious diseases, and as we've cut them out, we've started to grow older, and now we're seeing a whole new set of what is killing us now at old age. And now we're seeing cardiovascular disease, and as we cut that out,
into the new disease,
and as we go into the 150s or whatever is the next large area of longevity, what we're going to, maybe we'll find something else, which is the big problem.
Yeah, that's an interesting point, because even in the 1800s, I think 35 was old age, and that was the average age of death. And with things like sanitation, wash your hands, dentistry, childbirth improvements, half of women used to die in childbirth for most of human history. So it's only very recently where we've actually started living, not dying.
Beyond the infectious part,
So beyond the infectious part, that's right, and now we're encountering diseases of old age that we weren't really seeing before. So I like that idea of, because now people might live to 100, if we're living longer, we might encounter some disease we don't even know about.
Cardiovascular,
new problem.
Cancer's done, and now we're dealing with
some
something
new
else.
range of...
And I wonder if because as we see, even in the 30s, we still had cancer, we still had cardiovascular disease, but it's that it's increasing in proportion to what infectious diseases are now. And I wonder if maybe the next step isn't so much these biological ones that are within us or attacking from other parts, is that it's more of a society thing. And so we're seeing massive increases in mental health, and isn't that a huge social problem, really? We see it far more in impoverished regions. Poverty and mental health
is huge. Like suicide is one of the leading causes of death.
And it might be just as we're in the hundreds plus, it's suicide, it's mental health that are the largest killers. And
maybe that's what kills you at 150.
Maybe it's just social problems at that point.
Social problems are way bigger than they get resourced. Like the amount of money that's spent on the road toll, which I think is 3 million per year worldwide. I don't know what suicide is, but it's 10 times higher probably.
And even in terms of cancer, cardiovascular disease, mental health issues, and how they affect productivity or just people going to work, how they function in society. It's one of the most detrimental effects is just mental health problems and
that's an important space.
Mainly. So
how do you think learning more about DNA and the control of it will affect mental health?
I don't even know. So that's the thing, is that maybe it's more of a social problem there.
So it's socials beyond that.
So maybe we might solve all of these biological issues.
But we still have economic and social.
What is the...
So it's cultural overhangs. We're too slow. Our technology is exponential, but our social is linear, right?
Yeah, well, that's maybe why we're so scared of robots
and
too.
New technology.
So it's the anti-GMO. We're terrified of it because it's snuck up on people essentially, but no one understands it.
That gets me like legislating for things like editing genomes. Legislation's barely ready for gay marriage. This is a 1,000-year-old problem, and it's barely handling that. We've got this new age problem, modifying living things forever. And it can't even handle marriage, and
dealing with CRISPR-Cas9 and being able to word it properly. Especially when it's politicians that are creating these.
People don't even understand what it is, and we're going to try and legislate for it. When something easy to understand like gay marriage, and we still
we're still fighting.
It's so far behind. So what are we going to when a robot wakes up? If AI succeeds, because Google and Facebook and things, they've been putting in hundreds of billions of dollars into deep learning and neural nets. AI engines can capture images, learn to play old school Nintendo games at superhuman levels. Humans will never beat robots at chess again. AlphaGo beat Lee Sedol
Yeah,
in
we've
Go.
lost on the most complex board game we have.
Poker, which is the first like imperfect information game. All the AI games have been perfect information games so far. But just recently, an AI engine cleaned up World Poker Championships, so it bluffed, and it did all these sort of intuitive things, and it's sort of black box programming. Like we don't know how it comes up with its answers.
Yeah, it's coming out of the simple gameplay to human ideas and thoughts.
Yeah, so and the discussion's not had like society isn't ready for that.
No, so
but it's going to be on your doorstep, right?
There's few groups which are dealing with that in terms of what is like the moral implications, and how do we deal with it when they can think.
Like the robot rights.
Yeah,
and stuff.
and there's nothing. It's not
nothing's there yet.
No, so when it happens, we're going to be well behind. And with any tech we're coming out with, the CRISPR-Cas9, we don't have anything for it. It's snuck up, appeared out of nowhere,
and it's untraceable as
yeah.
well. I remember Sam Harris's quote where he's trying to relate artificial intelligence, and it's like if we received a message from an extraterrestrial species that said, "Dear humans, we'll be there in 50 years, get ready."
Yeah.
That's what AI is kind of like, where it might be possible, and if it might be possible, it might happen soon. And so that's the message we're getting, and we're just not ready.
Yeah, no one's reacting.
Awesome. But kind of crazy.
Yeah.
Alright, Sam, well, thank you for joining us on episode 1. And we'll see you again after your PhD, hopefully. And,
I'll be back just after that.
Sounds good. And thanks for listening. We shall see you for episode 2.